To start, let's review Mendelian inheritance patterns. Gregor Mendel was an Austrian monk who lived in the 19th century, and long before we knew anything about DNA, he noticed inheritance patterns in a variety of organisms, including the pea plant. He worked in obscurity for a long time until his revolutionary work was rediscovered, and his work fit with what became known about genes and inheritance and how that relates to the phenotypic traits that we see. So here we're going to look at simple traits that arise from a one locus, two allele system. And there are actually relatively few traits that are actually inherited this way, dry earwax of all things being one of them. But exploring inheritance in this way makes tracking genotypes from phenotypes fairly straightforward. In this example, we're going to look at pea color. So if we take a pure breeding green plant, which just means it always produces green peas when bred with another green pea plant, and we cross that with a pure breeding yellow pea plant, which just means that it always produces yellow peas when bred with another pure yellow plant, we get a first generation of offspring of peas that are all yellow. If we then cross those yellow peas together, the offspring from that breeding pair results in 3/4 or 75% of the peas being yellow and 25% of them being green. So how does this happen? Some, if not all of you, have probably used a Punnett Square, which is named after Reginald C. Punnett, who first described this approach to look at Mendelian inheritance. And a Punnett Square is just an organized way of predicting genotypes and phenotypes from simple crosses. So let's work out our predictions for genotypes and phenotypes for this particular experimental cross. So we know that we have a green pea plant. Let's represent the green allele with a small a, and we know that it's breeding true. And we have a yellow pea plant that is also breeding true. So we have a one locus, two allele system. So the two alleles that are here in this system are a capital A and a lowercase a. So this is the green pea plant, and this is the yellow pea plant genotype. If we cross those two, this is the Punnett Square example, we can see that we just fill in the boxes here. We have a big A and a little a, and we keep doing that. Big A, little a. Here we have a big A and a little a, and again a big A and a little a. And we know that this is the F1 generation, and they are all yellow. So the genotype frequency is 100% big A, little a. But what happens in the second generation when we cross the yellow pea plant with the yellow pea plant? Well, let's take a look. We know that we get 3 yellow plants and 1 green plant. So how does that happen? Well, if we're looking at the F1 generation, we know from our first cross that the genotypes of these two plants are big A, little a. So if we do another Punnett Square, we put the genotype up here of the one parent and the genotype over here of the other parent. And then we fill in the boxes just like we did before. So we can see that we have 1/4 are big A, 2 of the 4 are big A, little a, and 1 of the 4 is little a. So the genotype frequencies would be 25% big A, big A, 50% big A, little a, and 25% little a, which amounts to 25% green plants, but 75% yellow plants. And that's the phenotype frequency. But remember that a heterozygote, something with a big A and a little a, has a yellow phenotype, and we know that 50% of them are heterozygotes, and 25% are big A homozygotes. If we were asked to report the phenotype frequencies of the F2 generation, we would say there are 3 yellow to 1 green, or 75% yellow to 25% green. If we wanted to report the genotype frequencies, we would say that there were 25% big A, 50% big A, little a, and 25% little a. And we know that from the Punnett squares that we did.